Hello, this is Dr. Paul Cottrell, and I'm going to be talking about pseudouridine and how it is incorporated in the body and how they use pseudouridine for the messenger RNA platform. Okay, let's start with what is uridine. Okay, now uridine structure, chemical structure, is this. And pseudouridine is a similar structure. It has the same five-sided ring. But the six-sided ring are slightly different. Okay. So where are the differences? Now, we take, say this. So the differences you can see are your double bond here is no longer a double bond because it's incorporating a nitrogen. And you no longer have a nitrogen in this location, but you do in this location. Okay. Now, now, uracil is this structure here. Uracil is this structure. This is an H. Double bond. So you can see that this structure for uridine, right, follows the uracil, right? So uridine is this whole molecule. Okay, so really uridine is uracil with this 5-ring structure. Okay. Now, you can see that the reason why this bond is taking place, is due to this reactivity of nitrogen and the hydrogen. Hydrogen drops off, and then you end up being able to connect through enzymatic assembly. All right, so why is this important? Okay, so you have uracil is one of the nucleotides and it's incorporated in the DNA. So you have A, T, C, G, right? And then uracil can replace the T for RNA. So RNA doesn't have the T, RNA has the U. So this is the U, okay? So, and it needs that backbone, this 5-ring backbone. All right, so with that said, pseudouridine has this kind of like modified structure. Okay, so it's different than this is an H. You can kind of see that this is kind of like a rotated structure. All right, for uridine. All right, for example, this one here is here, right? This one here is here. This one here, 3 is here, right? And then this flips. This here, 4 is this one. And then 5 is here. Okay, so there's a rotation, there's a flip that's happening. Okay, all right, so now that's the differences between uridine, which is uracil with the 5-sided attachment, and pseudouridine, which is the rotation. It's really rotated. Okay, and you lose the double bond because of this attachment to the ring, the 5-sided ring. Okay, now let's talk about why is that important? Okay, so let's do a new screen here, and we're going to talk about different types of pseudouridine. Okay, pseudouridine can be incorporated into different RNA. Okay, and there's different types of RNA. There's tRNA, there are mRNA, there's ribosomal RNA, there's SN RNA, Okay, so why is this pseudouridine incorporated? Well, like I said before, when you look at the structure, this right here, it is it's actually more stable than the uridine structure. Okay. Okay, now, and what I mean by more stable, it adds structural integrity. Okay, so with the tRNAs are for holding the amino acid and transferring it to the ribosome for incorporation to make that polypeptide. The messenger RNA is the code. This is the code for making the protein or the peptide. And proteins are made from the amino acids. The ribosomal RNA is the machinery that's reading the messenger RNA and also binding with the tRNA and some other factors to transfer this to make the protein. Okay, so the RNA plus the tRNA is really making the protein. There's a little bit more complexity because of some transcriptional factors and what, but that's detail we don't need to go into. Okay, the snRA. All right. This is small nuclear RNA. Now, there's also microRNA. I don't know, people have heard of this microRNA. Basically, what's happening here is that you have small pieces of RNA, and these small pieces have like structure, right? And they might do something. They're attaching to the opposite for some reason, and you might have a longer structure, right? This is your micro snRA, and it can attach and stop some function or actually change the function of this RNA, this larger RNA, this larger messenger RNA. So there are inhibitory factors, and these kind of act like as a modulator in a way. Okay, it also can be used to break down splice. There's a whole bunch of things that these things are starting to be found to be doing. Okay. All right, so that's what these things are. All right, now let's talk about what is going on with pseudouridine. Now, now let me make a caveat here. This there are different types of pseudouridine. Okay, and I'll go into the one that is being used on the messenger RNA. I should add that to this slide so you understand. Now, the difference between the Caraco and Wiseman pseudouridine is what is called, what is called N1 methyl pseudouridine. Okay. Now, what the difference is this section here. Clean this up a little bit. Is this area right here, okay? On pseudouridine, which is endogenous, we'll have a nitrogen group and a hydrogen attached to the nitrogen. But what has been found is that you can take that hydrogen out, right? And you can actually attach a methyl group to it. And what will happen is now you have a methyl pseudouridine. Okay. So this is what's in the messenger RNA platform. All right, is this difference. Okay, but the regular pseudouridine at this position, is actually the regular pseudouridine that's endogenous is actually a nitrogen and a hydrogen. Okay. Okay, so with that background, now let's go to endogenous pseudouridine. Okay, now we have a structure that is something similar to this. Okay. All right, there are these hairpin loops. And you have the ACC sequence. And this ACC sequence holds an amino acid, some sort of amino acid. And this is where the codons are. Okay. And this is reading the actual messenger RNA. Okay. You know, and obviously you have machinery that's holding the ribosome. Okay. Now, there are areas. Now, this is a transfer RNA. And it will have AGCU, no T. Now, in certain areas, it helps to have the structure more stable when it has also a modified pseudouridine, okay? Or this, this pseudouridine. We're talking about endogenous. So this U can either be a regular U or, uridine or pseudouridine. Now, the way it is represented is with this symbol here. Okay. But this is pseudouridine. So there are some key areas for this structure here, to be more stable by using the pseudouridine. And those areas can be right about here. Oh, I don't want to do that. How do I go back? Right about here. Right about here. All right, so this will give it just enough structure so it doesn't break apart as well. So, you have the anti-codon, and you have the codon. Okay, so because these structures are here, this is leading to a better interaction with the RNA interaction with the rRNA, which is the ribosomal RNA, and the messenger RNA. Okay, so this is helping with this, for it to be more structurally stable and have the interaction with the ribosomal RNA and the messenger RNA. Okay, now let's talk about the messenger RNA. Now, pseudouridine can be found in messenger RNA. And it seems to be that it's affecting the stop codons. So we have three different stop codons. We have the UAA, the UGA, and the UAG. Okay, these are stop codons. Now, stop codons basically is that when you see the sequence, then the ribosome will basically fall off and stop making the polypeptide. So this stops the production of the protein. This stops it. So what this does, this pseudouridine with that change in structure, remember you have this 5 ring. Right? I'm not going to draw it completely. So you have the 5 ring plus the 6 ring. This is the pseudouridine. Okay. So this structure for the stop codons for either U or for the pseudouridine, and for actually U to C. They are promoting nonsense suppression. So nonsense suppression basically means so there's different types of kind of like mutations, right? And some mutations can be found or seen, and some are like hidden, right? All right. So what this will do is so you can have a mutation within, let's say you had in your messenger RNA code. You had in your messenger RNA code a U, G, G, A, T, and then a stop codon, U, A, A. Okay. So the stop codon is downstream. But let's say you had a mutation at this point here. And this UJA, this has a bunch of stuff too, right? Okay. Let's say this position here actually mutated and it turned into an A. Well, you would have UJA or UGA, right? UGA would be a stop codon. It would stop here. Stop. Right? And wouldn't reach to the downstream. Okay. So that's a mutation that's a nonsense. Okay, it stops prematurely. Okay. Well, this pseudouridine is preventing mutations in that area. So, for example, in our example here, let's say this pseudouridine, this uridine turned into a pseudouridine. Okay. The probability of this going into nonsense now has gone down. And this is part of the reason why. This is one. I'll explain it a little bit more with the vaccine, with the messenger RNA platform. But this is part of the reason why they were using the pseudouridine. this N1 methyl. Pseudouridine. Right. That's part of the reason. Another reason is that it actually slows down. It's this methyl pseudouridine that they used. It slows down actually the production of the peptide and preventing the slippage of the ribosome. Okay, now let's talk about ribosomal RNA. Okay, now they're different. There are two main features. There's a bottom feature and a top feature of the ribosome. All right, and different subunits. And different organisms have different numbers, right? There's a eukaryotic one and there's a prokaryotic one. And there are subunits to this. Okay. Okay. Now, and this reads the messenger RNA. And then it incorporates the tRNA that we just talked about. And when it has that loop in there, remember that loop that I drew? And it has that amino acid on the top. It has features. It has kind of like three cavities: a load category, like a transfer cavity, and then an exit cavity. And this area had, a tRNA leaving. Okay, it's exiting. There's a tRNA here that's getting this, and this peptide is growing over time as it's reading through the messenger RNA. All right, and eventually, so it goes from 1, 2, 3, right? Loads in, transfers the amino acid, grows the amino acid, and then the amino acid growing chain is transferred, and then the empty tRNA is exited out. Okay, this is part of the reason why the transfer RNA needs that pseudouridine, because of this kind of this interaction that's going on between the messenger RNA and the ribosome. There's a lot of stress. There's a lot of torquing that's going on during this feeding into the different cavities and the transfer of that peptide, that amino acid to build up that peptide. Okay, now, right, that's what the basic structure is. Okay, now what's going on with the RNA for pseudouridine specifically? So it's found, so you have the larger part of the RNA and the smaller part of the rRNA, the ribosomal RNA. So we'll call this small, call this large. And it's found in both. So the pseudouridine is found in both of them. Okay, so what's happening here is that the pseudouridine is providing structure. Right, you got to remember this. There's no way I can draw this, but this large and this small assemble onto the messenger RNA. But this structure is actually a RNA structure, and it's complex. It's there's no way that I can perfectly draw it, but it kind of folds on itself. Okay, and then this one will do something similar. Okay, but in some key regions, there are clusters of pseudouridine. And those clusters are in domain 2, 4, and 5. And what they do is they're stabilizing the interaction of the smaller and the larger to interact with this. And also, remember those cavities that I drew in here, over here, right? These cavities, right? That interaction to maintain the building of that protein that's coming out is also helped by the pseudouridine cluster that's in the RNA, the rRNA. And then the way this is, so it's not just the clustering. It's not just the clustering of the pseudouridine in some regions. But it's also, these clusters are to help for the attachment to the messenger RNA and to kind of hold the RNA, to transfer RNA in place and to allow for this peptide to grow. But it's also used to allow for this to properly fold. So, and again, there's no way I can draw it, but there's for the RNA, the rRNA, the ribosomal RNA, the pseudouridine does two things. It's helping with the RNA-RNA interaction. And that RNA-RNA interaction is the messenger RNA to the ribosomal RNA, and it's also the ribosomal RNA to the transfer RNA. Then there's also an RNA protein interaction, and that's this building of this peptide. But in addition, so that helps with the interaction, but in addition, it helps with the folding of the ribosomal RNA to its proper form. So pseudouridine helps with folding, helps with folding these complex structures. Okay. Now, again, this is the endogenous form. This is not the methylated form. Okay, so, all right, now what we want to do is discuss now. I'm doing this on the fly, so I'm just, I didn't prepare this at all. So pardon me if it seems discombobulated. All right, snRNA. Okay. So they can also have the pseudouridine. Okay, now we have, I don't want to do this. Okay, we have a messenger RNA. Right? And we have exons and introns. Exons, introns. Exons, introns. Okay. And this can be folded and spliced in a way where we only have exons. So exon 1, intron 1, right? Exon 2, intron 2. We want an exon 1 plus exon 2. So that means that we're kind of splicing. We're splicing this. And we're splicing this off. So we only have this and this, okay? So the snRNAs help with this splicing. Now, where the pseudouridine comes in is that I'm not really sure what the structure of this small nuclear RNA is, what it actually looks like. Okay, I think it looks something like this. Okay, it has kind of like this feature. Okay, because it's not just, remember, you can have RNA that's kind of like that. You can have hairpin type RNA, right? And then it's opened up, and it helps to fold certain of these structures kind of together, right? That's one kind of RNA shape, all right? But we normally just write it like this, right? But this thing can kind of fold on itself, right? There can be hairpins and stuff. Well, this small nuclear RNA looks something like this. So it gets a little involved, the way it looks. And it's like folding on itself. Okay. It has this kind of feature. Now the circles are areas where they're not bounded together. This with a complement. These have a complementary sequence. And this structure interacts to help with the splicing to get the proper protein. Should draw it. Proper protein. Okay. Now, if you had intron or exon, let's say exon 3, right? you can have a splicing where you're making E1 plus E3. So this is an E1 plus E2. And this protein, different type of protein, maybe E1 plus E2, E3. So this helps with this splicing function, okay? Well, pseudouridine in certain areas are going to add to stability, similar to the other features that's going on with the RNA, the messenger RNA, and blah, blah, blah. So in certain areas, you're going to have this RNA interaction, and you're also, it'll help with that, and then it'll also help with the RNA protein interaction. Okay, so now where specifically they are, I don't know. I mean, I'm not really sure where the binding of the I would have to study the snRNA at the molecular level to understand, you know, where are the structural components, because I'm not sure where, you know, where in this, you know, the main docking areas are. I'd have to study that. I'm not really sure. And then again, the folding, it helps with folding. The pseudouridine is helping with the folding of the snRNA. So again, pseudouridine interaction of the RNA to RNA, interaction to RNA and protein, and folding of the small nuclear RNA. Okay. Okay, so now in this video, I went through the structure of uridine, uracil, and how the pseudouridine has an NH at this area, but that hydrogen leaves, and then there's a methyl group that's added for the methyl pseudouridine that's used in the messenger RNA platform. Then I went and explained to you the differences between the tRNA, the messenger RNA, the ribosomal RNA, and the small nuclear RNA, and did a little bit on the microRNA, and basically what those functions were. Then I discussed the tRNA and where the interactions are for the pseudouridine to allow for better interaction with the ribosomal RNA and the messenger RNA. Then I went and described how pseudouridine can be used to prevent the probability of a nonsense mutation. It's preventing the production of a stop codon. Okay, and this is part of the reason why the methyl pseudouridine that's used in the messenger RNA platform was used, but it's also to help to slow the production down. But that's not, those aren't the only two reasons. The methyl group, and I'll say this in a different video also, this methyl group here actually flies underneath the radar and prevents the the immune system from detecting it as a foreign product. Okay, now, then I went into the ribosome RNA and how the pseudouridine endogenous helps with the RNA-RNA interaction and the RNA protein interaction, and how it helps with the folding. You know, a key piece here is how the small ribosome RNA and the large ribosome RNA are helped structurally for folding with the pseudouridine. Now, here, the small nuclear RNA, helps with the splicing, right? Now, it also has, RNA interaction, stabilizes the RNA interaction, the RNA protein interaction, and also the folding of the small nuclear RNA. Now, exactly where, I'm not sure. Is it here? Is it here? Is it here? I don't know. I don't know how this is docking to how the small RNA is docking to it. I'd have to study that a little bit more, exactly what the binding domain is, but I don't really care, actually. But all right, and then okay, and then in the next video, what I'm going to do is I will go in a little bit more detail on the endogenous and the methyl RNA. The methyl pseudouridine on the messenger RNA platform. So hopefully, this is going to be probably a 2-video series, maybe 3-video series, depending on how long it's going to take me to do this second video. But at least this is a good primer to understand the purpose of pseudouridine endogenously. Again, endogenous pseudouridine doesn't have the methyl group. It has hydrogen attached to this nitrogen. That's the endogenous. But the methyl pseudouridine, which is used in the messenger RNA platform, does have that methyl group attachment to the nitrogen. Okay, so thank you for watching this video. Hopefully this teaches you a little bit of the biochemistry of it and how it interacts with key machinery within the cell, such as the tRNA, the ribosomal RNA, the small nuclear RNA, even in the messenger RNA, and is a good primer as we dive a little bit deeper into the synthesis of this actual protein. Now, and I'll also bring up the cancer stuff too. I mean, there's some interesting things that are going on with pseudouridine incorporation to stabilize proteins, because in cancer you have a lot of fusion proteins, and you have actually a chromosome that's breaking down. The chromosomal integrity isn't there. That's the reason why it goes into cancer, right? But if it knocks out some pathways, then what will happen is what's interesting with some cancers is that it actually has an upregulation of this pseudouridine endogenous and stabilizes these structures. Even though you have a breaking down of the genome and more fusion proteins, you're actually stabilizing some of these interactions that I just discussed. So, and that may be part of the reason why, from an evolutionary point of view, cancer is, can survive in the body and break away from the natural immune system from clearing it out. It's not just having its apoptosis pathways being knocked out, but there is this interaction of pseudouridine stabilizing the molecule. All right, so that's just a little bit of a, I'll go in a little bit more detail on my thoughts on that. So hopefully you've learned something from this. Apologize for my drawings. I don't draw very well, but these sketches, especially while I'm talking. But please go to my store, the-studio-reykjavik.com, and purchase the products that I offer. I mean, I have a lot of really, really good health products. Like, for example, let's see if I can share my screen here. Sometimes on the iPad, this is part of the reason why I don't like doing the whiteboard so much, is that, sharing on the iPad isn't as streamlined as sharing on the laptop. And, as I get older, I just want a simplified life. I'm tired of this, tired of technology, really. Okay. So here is my website, and you can go to the store by just clicking store, and then I have many different products. I have the nano silvers, the C60, the Boomer products, the all natural deodorants and stuff. But please go to the store, the-studio-reykjavik.com, and purchase the toothpaste that I have. There's no fluoride in it. This is a very, very high quality toothpaste. It's going to be the best toothpaste that you ever tried. It'll neutralize pathogens. Because it's neutralizing pathogens, it's going to be removing preventing, additional plaque, because plaque is the buildup of because of the pathogens that are in your mouth. And by reducing that gum inflammation and reducing the plaque, you're reducing the chances of the cardiovascular disease. In addition, I have liquids. I take Max 35 every day with a teaspoon, and if I'm not feeling well, I'll take a tablespoon of it, or two tablespoons. But that's a great product to neutralize pathogens. Basically, what you do is you swish it in your mouth and you gargle with it, and you can just swallow it. And this is keeping that mucosal lining within your upper cavity in proper health, to prevent some sort of barrier disruption. Silver gel, that's a great product. What I do is I put that on my hands and around my face and nose and ears to neutralize pathogens. I have the nano soaps. I have the lozenges. I have right now in stock the sweet menthol lozenge. Probiotic. This is in the capsule form. I also have a powdered form by Boomer. I have a great multivitamin that you should try. So, some very good, high quality structural nano silver products. The C60 is a great, very strong antioxidant. All right, it's in coconut or avocado oil, but this compound just soaks up those reactive oxygen species, and when you do that, you're reducing the cellular stress on you're reducing the cellular stress because it's soaking up those charged oxygens. when you're doing that, this is going to increase the mitochondrial health of the cell, and you will have more energy because the cell is producing the ATP. So what's really important is just strong antioxidants will help with your body getting into homeostasis, because that chronic inflammation and all that, it puts a lot of stress on the cell. So I have it in 2 ounce, 4 ounce, and 8 ounce. Again, in coconut oil or avocado oil. For the Boomer products, I have D3 that is very good in being able to apoptose cells. It's also a cofactor for gene expression. So it's a very important supplement. On top of that, it helps with the absorption of calcium. Vitamin C, obviously you don't want to get scurvy, right? And then omega-3, great for cardiovascular health. Ashwagandha, great way to control blood glucose levels. Good night formula, which is tryptophan and melatonin, a great way to maintain that REM sleep that you need to solidify memory and to just get your body actually is healing when you're sleeping. But if you're not sleeping properly, your body's not healing properly. The probiotic that I have here is the powdered form. It's a great way to just put it in a smoothie, or, for me, I put the powder in my hummus. Resveratrol, a great antioxidant, is synergistic with C60. The B complex, your B vitamins are cofactors for processes for metabolism. In addition, if you don't have enough of these cofactors, you're not going to have proper electron transport chain function within your mitochondria. And, being low on your B complex can be associated with anemias. Turmeric is a great, not only an antioxidant, but it brings down that that inflammation. Remember what I've been saying? I've been saying control your reactive oxygen species by neutralizing them, with the C60s, with the turmerics, with the resveratrols. But turmeric also brings down that inflammatory response. When you bring down that inflammatory response, that's another way to reduce the reactive oxygen species, because there's less chance of creating them because there's less inflammation. Zinc and magnesium are important cofactors. Lignans, great for hormonal balance and support. Collagen, it's great for the skin and to help with the cartilage, especially if you've been like a runner for a long time and you're starting to feel that osteoarthritis. Stop running so much. Start walking is probably some of the best things. Swimming is a really, really good exercise. But just kind of let that cartilage and those tendons and those ligaments start to heal. It takes a while, if you've been injured, but collagen is one of those components that are needed to kind of help rebuild those joints. But this product is a great product. And if you take it with the vitamin C and drink 64 ounces of water, you're going to see an improvement of your skin. And then if you take it with the C60, which is the strong antioxidant, and the resveratrol, which helps with getting rid of apoptose cells, you'll see an added benefit in your dermal layer. The digestive enzyme complex, as we get older, or if you are really into training for something like a marathon or whatever, you need to be able to break down those carbohydrates, those proteins, and those lipids, and reduce that load on your pancreas. So this can be a supplement for individuals that are in the gym all the time, working out, and need a recovery and need to be able to fuel quickly. But it's also really good for individuals that are getting older and they don't want to you don't want to put so much load on their pancreas. As we get older, our pancreas does start to wear out. So this helps with that, to be able to break down those lipids, proteins, and carbohydrates that we eat. The Clarity Factor is a great product that helps to get rid of that brain fog and allows for you to be able to concentrate. I take this twice a day. I take it in the morning and I take it in mid-afternoon. So you take it, that first week, and you're going to see a major difference in your ability to concentrate and to be able to solidify memory. And when you are clear and you're not, in this fog, you're going to just have a better day. And on top of that, you're going to have better sleep. And I have these all natural deodorants that Rainbow Herbals makes. The even better bar is for muscle pain, for cuts, scrapes, bruises, burns, minor burns. What will happen is you'll heal faster. What's amazing about this product also is that even if you don't have anything wrong with your skin, if you start to apply this on your skin on a regular basis, you'll actually start to see a health benefit of the skin, and it'll be smoother and healthier. So you can use this as a skincare, health skincare product to improve your skin, or as more of a muscle ache. You'd be amazed that, you get a muscle ache, you put this thing on, and it actually goes away. Now these ingredients are high quality Himalayan essential oil that's been put into a bar form. So easy application, very, very high quality. And in addition, I have the citrus deodorant and the peppermint lavender tea tree deodorant. So try those out, all natural, very high quality. Again, essential oils from the Himalayas put into a bar form. And, this, you'll notice, added benefits because, it doesn't have the aluminum in it, and it doesn't have, all these, funky chemicals that you would get from, let's say, a Procter & Gamble product. Now, lastly, for the store, I have these different anti-aging boxes that I curated. And, at different price points. And basically what it is it's like a kit. So, if you, want to get, let's say, a Max 35 and a C60 and, D3 and vitamin C and toothpaste and the deodorant and the probiotic, then the advanced box would be a good pick for you. You can also buy these products individually, but if you wanted to start a beginner's box for, let's say, skincare, great way to do it is get the collagen, the vitamin C, and the gel, the silver gel, and that will help. The key here is also to drink the 64 ounces of water. If you want to get an anti-Fauci box, that would have the Max 35, the vitamin D3, the magnesium and zinc, the turmeric, and the ashwagandha. So, I got all these different curated boxes for you for just a one click to get, the products that you need, especially if you're a beginner. Or you can just buy the products individually. Go to the store and buy the products individually. But hopefully this will, help with your health. Now, in the home page here, at the very bottom, you can donate through Stripe or PayPal to help support my news coverage. You can also donate on Buy Me a Coffee. All the links are in the description of this video and all my videos. So please help by donating or going to the store and buying the products that I offer. Very, very high quality products that you get. And you will notice in a very short order the health benefits of following this protocol, this anti-aging protocol that I've been saying for a long time now, for years actually, and something that I've been doing since I was 16 years old, and improving on it as different supplements came to the market. Now, please also subscribe to my channels. I have 3 YouTube channels. So please subscribe. The links are in the description of this video and all my videos. And also subscribe to my Patreon, Brighteon, and Rumble channels so you can see the content. A lot of the things that I bring up, I cannot keep up on YouTube because it's talking about, sensitive issues. But if you, listen to my premieres and, go and watch the videos that are stored on Brighteon, Bitchute, and Rumble, you'll be able to, see that content that's been heavily censored on YouTube. So it's very important that you subscribe to all 6 channels. And if you could, please subscribe to my Patreon channel for a nominal fee that you can help support my news coverage. Thank you for supporting my work. Thank you for being a loyal follower and listener, and hopefully you're learning something as I do these videos. It takes some effort to do these things and some time, so hopefully you appreciate the effort that I put into it. But I do appreciate your support and the individuals that are going to the store and listening to what I'm saying about this anti-aging protocol and how to improve your health and boost up your immune system. Thank you for listening and have a nice day.